start talking about clinical imaging, in particular clinical imaging of actinium in a PET scanner. So it's the last day of the conference, so I'm sure that you're familiar already with theranostics and why imaging is critical for this modality. So the way that this is typically implemented in the clinic is via surrogates, so surrogate imaging with PET scanners. So that, say, you inject a PSMA PET and then you see where it goes, and then after that you inject the therapy. Or you can do direct imaging of the therapy itself with SPECT scanners. So the problem with these two modalities is that the first one, PET, only works with positron emitters. So you cannot do imaging directly of actinium in principle. And then SPECT scanners, we've been seeing that they have low detection efficiency and give you higher noise. So I would like to propose an alternative, which is direct imaging of actinium with a PET scanner. And this is via not PET reconstruction, but time-of-flight CGI reconstruction. So let me talk, and this is in order to capitalize on the higher detection efficiency and lower the noise that you can typically get with these type of scanners without collimators. So let me talk about this modality, time-of-flight cascade gamma rays imaging. So the main ingredient is a gamma cascade. So you need a fast gamma cascade, very fast, typically sub-nanosecond. And it turns that in actinium we have a sub-picosecond gamma cascade. So it goes through the thallium branch. It's only 2% branching ratio, but most of the decays of thallium have this gamma cascade. It's a triple gamma cascade. We're gonna just focusing on the ones that I'm highlighting in yellow there. So it's a very fast cascade, sub-picosecond, with energies which are very different from PET energies, 511, right? See, there you go. So how do we exploit this gamma cascade? So the idea is the following. So say you have a point source in a time-of-flight capable scanner. You have the first gamma that is emitted at a certain time and is detected at time 1, and then you have the second gamma that is detected at time 2. So in a time-of-flight capable scanner, you measure the time of flight, which is the difference between T2 and T1. So that part is constant. So since that part is constant, that's defining you a hyperboloid, because the subtraction of the gamma 2 arm and the gamma 1 arm is constant. So that hyperboloid is not a shell, it's a probability function, because which thickness depend on the time of flight of the scanner, like the CTR, the time accuracy, and also on how fast the cascade is. This cascade likely is very fast. So this gives you a modality that allows to image non-positron emitters in PET scanners. And just to illustrate a particular case, this is with synthetic data of a point source. If you overlay many of those hyperboloids, then you recover the location of the point source. In practice, we use an OSCM framework that we wrote in order to accommodate time-of-flight CGI that we call POSM. It's written in Python. I don't have much time to talk about it. Just gonna show some results. So the first step is to make sure that we can detect this gamma cascade. Right? If you don't get the data, we cannot do this reconstruction. So we partnered up with Siemens. They were very nice to open up the scanner that we have based at UCSF. So this is in an actual clinic, the Biograph Vision 600. They put it in research mode, and then they vary the gain of the channels in order to accommodate the energy, especially the very high energy of the second gamma ray. So the time window was accommodated to 300 kV to 1.6 MeV. And then we acquired the least mode data and we run our reconstruction. So first, if you just run the scanner in this mode with no source, this is what you're gonna get. What you see there is the lutetium-176 background. So you still have this background, but fortunately on the energy region of interest, you have nothing. There is no signal there. But once that you put the source, so we started with a 5.8 megabecquerel actinium source located at the center, then you see that gamma cascade coincidence, right? And then we measure a CTR, time resolution, of 205 picosecond, which is a little bit better than with PET. The theory is because of the higher energy gamma ray, of the second gamma ray. Sensitivity was 75 give or take, counts per second per megabecquerel, which corresponds to a detection efficiency of 0.35% when you correct by the branching ratio of thallium. So we're in business. It looks like we can see this. Can we do imaging? The answer is yes. We did imaging of this point source. The spatial resolution that we measured was about 2 centimeters, give it or take. So the spatial resolution is direct proportional to the time of flight of the scanner. So the time of flight gets better, then your resolution gets better. This is slightly different than PET. The z-axis spatial resolution is a little worse. It's 4 centimeters, and this is because of the hyperboloids. Like the intuitive way of thinking about this is that the hyperboloids, because the scanner is very short, it's just 26 centimeters. The hyperboloids are very parallel to the z-axis, so then you have that elongation on the z-axis that we'll be seeing through the images. We want to talk about low activity imaging, so we did a low activity phantom imaging of 2 vials. We could distinguish both vials. We did a multi-vial imaging in order to characterize the linearity, whether this modality is quantitative or not. The answer seems to be yes. It's very linear, so we can do quantification. And the limit of detection for this particular experiment with these vials and for a 20-minute scan in this scanner was about 5 kilobecquerels. So things were looking good, so we filled this phantom with actinium at a low concentration, 44 kilobecquerels per milliliter. We ran a 45-minute scan in the PET scanner, and then we could recover the image. So we just fill the 4 spheres I can see there in a non-radioactive environment. We also compare with SPECT, in this particular case with bismuth. So I was a little bit surprised that we could see actually good images of SPECT, to be honest, in the literature out there. It's a little bit noisier, but we could still see something with SPECT as well. When you compare the sensitivities, you see that the sensitivity of CGI is 26 counts per second per megabecquerel, while the SPECT one is a little lower when you correct by the scattering background. The detection efficiency is about a factor of 20 more, right? Because remember that the branching ratio of thallium is just 2%, while the branching ratio of bismuth is 25%. So when you correct by that, you get a much better detection efficiency or CGI. And then we compare this SNR in order to be quantitative, and we see that we can see an SNR that is larger than SPECT. I don't mean this to be a systematic comparison between CGI and SPECT. I'm just showing this in order to have some sense, right, of what we are seeing here and what is happening with this modality. So it looked like we could do imaging of actinium clinical phantoms in a PET scanner. So we just went ahead and piggybacked on Dr. Hope's clinical trial that is running at UCSF. He's injecting actinium PSMA in patients. So we modified the protocol in order to do this extra CGI scanner. So the protocol, I'm going to flash very quickly here, is the following. We did pre-therapy, regular PET, in which we inject PET PSMA. We confirmed that the patient is eligible for this therapy. We injected 8 megabecquerels of actinium PSMA. We imaged it according to the protocol, 24 hours post-injection in SPECT, following just the vendor's recommendation with a high-energy neural pulse oximeter for an hour. We imaged all the critical energies or relevant energies. And then the patient came back 5 hours later. The patient for this trial was really a hero. He sat in 3 different, 2 different systems, three times. He came back for an hour of a CGI scanner. So this was just, again, a pilot experiment with a single patient, and this is the image that we obtained. So this is CGI CT image of the prostate bed, where we confirm and verify that the activity was in the prostate, or close to the prostate bed. So this is the CGI image, which I would like to recall is at thallium-209, and this is the pre-therapy PET image, where you can see that there is a very good one-to-one mapping between the pre-therapy and post-therapy. And just a little philosophical regression here. We're doing pre-therapy and post-therapy of PSMA PET and actinium PSMA in the same system, which I think is could be interesting. And then this is the comparison with SPECT without attenuation correction and with attenuation correction. I didn't say we are not applying attenuation correction because we don't have it in the algorithm yet. So here I'm showing kind of like the whole picture. We also can see some uptake in SPECT, as you can see there. SNRs for, again, one particular patient. This is not a systematic study, it's just a pilot study. The SNR seems to be higher for CGI. So just wrapping up, I'm over my time, so I'm gonna leave you here with the conclusion. We did actinium imaging, direct actinium imaging in a PET scanner, so that seems feasible. The clinical value of this modality will be given by future clinical trials. I would just like to thank especially the clinical team. They were very helpful, and Dr. Jan Joseo, who paid for dinner this day after the experiment. I don't know if he's around, but thank you very much.